Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Think for a moment about picking up a new hobby, maybe learning guitar, or trying a really complicated recipe, or even just getting your head around a dense news story. You don't really think about it consciously, but your brain is doing something pretty incredible, taking all that new stuff in and somehow storing it, making a memory. How does it actually do that?
Yeah, that's the big question, isn't it? Neuroscientists have been chipping away at that for ages. And, well, there's this fascinating recent study just published in Science that's uncovered some really fundamental things about how our brains learn, right down to the level of single nerve cells.
Neurons.
Exactly. And that's what we're digging into today. This research comes from that Science article by William Wright and Takaaki Komiyama, and it was also covered in a health news piece this week, April 18th, 2025.
Right.
So our mission really is to understand these new rules they found, how neurons actually encode new information. And then what does this all mean, for understanding the brain, maybe future treatments, even AI? Absolutely. It's got some potentially big implications.
Yeah.
But maybe first, we should quickly recap the basics, just so we're all on the same page about how neurons work.
Good idea. So the brain's packed with billions of these nerve cells, neurons, and they talk to each other using electrical pulses?
That's right. Tiny electrical signals. They travel down the neuron and then get passed on to other neurons across these little junctions called synapses.
Okay.
And each neuron has these branchy bits called dendrites that receive signals from lots of other neurons. The main part of the neuron then kind of adds up all these incoming signals. And if there's enough excitement, enough input, bam, it fires off its own electrical pulse. And it's really the coordinated activity, this collective firing of specific groups of neurons that represents, your memories, your thoughts, your experiences.
Right. So memories aren't like a file in one spot. They're more like a pattern of activity across a network. And the long-standing idea, the main theory, has been that learning happens because the connections, the synapses, change strength. Some get stronger, some get weaker based on your experiences.
That's synaptic plasticity.
Exactly. Synaptic plasticity, the idea that connections change strength with experience. It's been central to neuroscience for decades. But there's always been a huge puzzle, often called the credit assignment problem.
The credit assignment problem. Okay, what's that?
Well, it's about figuring out the specific rules the brain uses. How does it decide which of the literally trillions of synapses need to change strength? And crucially, when do they need to change during learning to store the right information? It's been a major mystery.
Right. Which connections get the credit or blame for a learning outcome?
You got it. And that's what William Wright and Takaaki Komiyama were trying to get at. What are these fundamental rules?
Okay, so how did they actually go about tackling that? What was the experiment?
It was pretty sophisticated. They managed to monitor individual synaptic connections inside the brains of mice while the mice were actually learning something new. Wow, individual connections. How?
They used these really cool genetically engineered biosensors. Think of them like tiny little lights, molecules that they put into the neurons.
Right.
And these biosensors literally light up when there's neural activity or when a synapse is active. So the researchers could actually see what was happening in real time at specific synapses while the mice learned.
Incredible. And what was the task the mice were learning?
It was fairly simple. The mice heard a specific sound cue, and they had to learn to press a lever to a certain position to get a reward drop of water.
So watching the brain wires change as the mouse learns. Sound means press lever this way, get water. What did they find? Did it confirm the old neurons that fire together, wire together idea?
Well, this week it's really interesting and pretty surprising, actually. The traditional view, those Hebbian rules, fire together, wire together, kind of implies a uniform process for a neuron. If the neuron's firing a lot during learning, you'd sort of expect all its input synapses to generally strengthen.
Makes sense.
But that's not what Wright and Komiyama saw. It was quite different.
Oh. So what did they discover?
They found that different synapses, even on the same neuron, but on different dendrites, different branches, they were actually following different rules for changing their strength.
Wait, different rules on the same cell?
Exactly.
Right.
Some groups of synapses did follow the classic Hebbian rule, consistent firing leads to strengthening. But other groups of synapses on that very same neuron behaved totally differently. Their changes seemed independent of the neuron's overall firing rate.
That's wild. So a single neuron isn't just one learning unit. It's got different parts learning in different ways.
That seems to be the key finding. Neurons are apparently using multiple, distinct learning rules at the same time across different sets of synapses on their dendrites.
Like multitasking.
Precisely. That's the term the researchers used, neuronal multitasking. By having different rules operating on different inputs coming into different branches, the neuron can process and represent information in a much more nuanced, more precise way.
So it's not just one simple calculation, it's like parallel processing within a single cell.
Kind of, yeah. Wright and Komiyama themselves said they were surprised. It suggests neurons are way more sophisticated in how they learn than we maybe gave them credit for.
Okay, this is a fundamental shift then. If neurons are multitasking learners, what does this mean for, say, understanding brain disorders, things where learning and memory go wrong?
Well, it could be really significant. We know many brain disorders, things like Alzheimer's, but also psychiatric conditions, involve problems with synapses. Connections might be too weak or too strong, or they don't change the way they should.
Right.
So if we now have a better blueprint of how synapses normally change during learning, including this diversity of rules, it might give us crucial clues about what's breaking down in these diseases and potentially new ways to fix it.
The news report mentioned depression as an example. How might this connect?
Right, so the thinking there, as the researcher suggested, is that maybe in depression, some synaptic connections get excessively weakened, particularly in brain areas involved in pleasure and reward.
Okay.
If we understand the specific rules that normally govern strengthening and weakening in those circuits, maybe we can figure out why they're getting too weak in depression and eventually find ways to specifically target those rules to kind of nudge them back towards normal function.
Ah, so much more targeted than current approaches, perhaps.
That's the hope. As Wright and Komiyama put it, understand the normal process is key to figuring out what's abnormal and how to intervene.
Makes sense. And what about the other angle mentioned, artificial intelligence? How could this influence AI?
Yeah, that's another really interesting potential outcome. Artificial neural networks, the basis of a lot of AI, were originally inspired by the brain, but in a very simplified way. The learning rules they typically use are uniform across the whole network and often, let's be honest, not very biologically plausible. They don't really work like real neurons seem to.
Right, much simpler.
So this discovery that even single biological neurons use multiple different learning rules could be a real inspiration for AI designers. Maybe building AI with more diverse, more brain-like learning rules could lead to systems that learn more efficiently, or are more robust, or maybe can handle more complex problems.
So smarter AI by making it more like actual brain wiring.
Potentially, yes.
Yeah.
The researchers definitely see potential there for creating more advanced, maybe more powerful AI based on these biological insights.
It's amazing how a finding about tiny connections in a mouse brain could ripple out like that. But you mentioned it's still early days. What are the big caveats or limitations here?
Oh, definitely. It's crucial to remember this is a big step, but it's still just one step. The researchers are very clear. We're a long way from translating this directly into new treatments for people.
Okay.
And there are still big unanswered questions, like they found different rules on different dendrite groups, but why? What tells a synapse you use rule A versus you use rule B?
Right. What controls the rule selection?
Yeah, exactly. And how are these different learning processes coordinated within the neuron? We don't know the mechanisms behind that yet. That's what future research needs to figure out.
And I think you mentioned that while this multitasking clearly boosts the neuron's ability to store information.
Yes, it increases the information encoding capacity. That seems clear.
Are there other consequences? Maybe trade-offs?
That's another open question. The researchers point out that other potential effects or properties of having these multiple learning systems in one cell aren't yet understood. There could be downsides or other benefits we haven't even thought of.
So more research needed, as always in science.
Absolutely. There's still a lot to uncover about how exactly the brain pulls off this incredible feat of learning and memory.
It really is astounding, though, just thinking about that complexity, that flexibility happening constantly inside our heads at such a tiny scale. Makes you appreciate the machinery.
It does. And again, the researchers themselves, right, in Komiyama, express that surprise. The sheer flexibility, the multitasking nature of these individual cells was not what they necessarily expected. They see it as potentially fundamental to how neurons handle the complex information streams they receive.
So thinking about this discovery that even at the neuron level, there isn't one single rule, but this diverse, localized set of rules working together. It makes you wonder, doesn't it? What other complex systems out there might work the same way?
That's a great point.
Maybe things that look uniform or centrally controlled from the outside actually rely on lots of different local strategies working in parallel to achieve something remarkable. It definitely makes you think differently about learning and maybe complexity in general.
It really does. It encourages us to look for that hidden diversity, that underlying sophistication, even in things we thought we understood. A beautiful reminder of the complexity hidden within the natural world.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.